Hi, everybody. Welcome back again to our next live webinar. Today, we are celebrating the World Ocean Week. And to all audiences, good morning, good afternoon, and good evening from different parts of the world. So I'm glad to have invited our students panelists, and we'll be talking about topic bite size. I mean, it will be a bite size student edition, and it's focusing on marine biodiversity. Okay, I'm glad to have today with us Aiden, Shikong, Daisuke, Jenny, Lionel, Nicholas, Samantha, and Sam. Okay, but sorry for a bit of delay today. There's some overrun from the previous panel. So without further ado, I would like to start the topic as soon as possible. I think a lot of people is waiting for us to share more about marine biodiversity. So Sam, over to you, please.
Okay, hi everyone. A big thank you to the edX team for the opportunity to share Singapore's research on this international stage and for me to moderate this discussion. So this session to me is very special because we have a group of younger scientists who are equally driven and passionate in marine research and conservation. So all of our guests here, they are PhD students from the National University of Singapore who are all very involved in interesting projects, which you'll get to see later. So we have Lionel, Nicholas, and Aiden from Reef Ecology Lab, Jenny, Daisuke, and Samantha from Experimental Marine Ecology Lab, and Chikong from Tropical Marine Science Institute. And today we'll be talking about marine biodiversity, which is the different kinds of living things that you can find in the ocean. And marine ecosystems, they provide us with a lot of goods and services like tourism, coastal defense, and food resources for many people. However, in light of climate change and the impacts of human activities, we are starting to see some changes in the ocean, right? So for instance, the marine environment in Singapore is very challenging. It's heavily sedimented due to extensive coastal development. So what does it mean for our marine biodiversity? What kind of marine life can we find in Singapore? And what are the local scientists doing to actually mitigate biodiversity loss? So today we will find out in this session. So before we start formally, each panelist will have about 5 minutes to share your work. So for our audience, keep those questions coming throughout, and then we'll answer all the questions after the panelists are done with their sharing. Okay, so without further ado, let's start with Lionel. So while Lionel is setting up his screen share, I will introduce him. So Lionel is broadly interested in coral reef ecology and marine conservation. So his current research focuses on the ecology and management of urbanized marine environment, as well as the development of strategies to enhance coral restoration outcomes in Singapore. Okay, Lionel, go.
Okay, can you hear me?
Yes.
Okay, so thanks Carter and the ADEX team for having us, and thanks Sam for approaching us to do this series of slides. So my name is Lionel, and at NUS I do research on ecology and management of urbanized marine environments, and I'm also interested in the restoration of coral reefs. So why coral reefs? Because coral reefs, they, as Sam has eloquently summarized, provide us with a lot of ecosystem services. They are a natural heritage. They are also homes to a quarter of the world's marine species. And therefore, losing them would definitely be very disastrous for all of us. So for today's talk, I'll be focusing more on the restoration in the context of Singapore. But to do that, we have to look at Singapore 50 years ago and compare that with now. So for Singapore, as most of you know, there's been a lot of land reclamation going on. And as such, we have increased our land area by more than 20% since the 1950s. And certainly the coral reefs are affected. There's been a lot of heavy sedimentation in the water, which smothers corals. The water is also very turbid, which cuts off the light needed for the corals to photosynthesize. This has resulted in the reefs becoming more shallow compared to decades ago. The reef substrate in many areas is also very unstable. There's a lot of rubble, so coral larvae that recruit on the rubble are at risk of dying when the rubble gets shifted around. So when you add all this up, it's no surprise that Singapore's coral reefs have decreased in total area. In fact, more than 60% over these past 50 years. Now, although we have some good news periodically, like mass spawning events every year, we also have observations of corals recruiting on artificial structures such as seawalls. This is not enough because corals, they grow really slowly, a few centimeters a year, and therefore the rate of recovery by themselves would definitely be outstripped by the rate of degradation. And this is where active management steps like restoration comes in. So restoration aims to put back the structure and function of the degraded of the reefs that have been lost. So for Singapore, we are basically looking at two main approaches. The first one looks at improving the quality of the reef substrate. So many of you are familiar with artificial reefs and reef balls, but in Singapore in the early 2000s, members of the Reef Ecology Lab put down fiberglass artificial reef structures at various reefs. And in a few months, you started seeing organisms colonizing these structures. In about 2 years, the juvenile corals are visible to the naked eye. And in the mid-2010s, when we went back to visit these structures, we were pleasantly surprised to see up to more than 119 species of reef organisms using these structures for food and habitat. You know, even damselfishes laying eggs on walls, urchins grazing on walls, and cuttlefishes doing whatever cuttlefishes do. And a few years later, in the late 2010s, we also noted that coral cover on these structures was quite high. So all this tells us that restoration is a long-term process. It might take decades to even see results, and therefore it should never be a short-term project. So the next approach is that we directly increase coral cover by transplantation. Before that, we use the concept of nurseries, like plant nurseries, where you grow saplings in plastic trays until they are large enough to be transplanted out or sold. For corals, instead of trays, we build tables. And these tables are designed to allow sediment to fall through so it does not accumulate and kill the coral fragments. And then we populate these tables with coral fragments, which we collect from the reefs. And these coral fragments are actually your loose coral fragments that otherwise would die if we do nothing to them. So might as well give them a chance of survival. So what follows then is an intensive period of maintenance and monitoring, because we really need to know over time how the coral fragments are doing, whether they're surviving, whether they're growing well. We also become gardeners where we have to remove algae from smothering them. And as they grow to a certain size, we transplant them out to target sites using marine epoxy, which is an inert substance that cures in usually half an hour. And then, of course, we monitor them over time. So with different coral species, we naturally have different responses for this particular type. From a tennis ball sized fragment, it takes years just to reach a deflated rugby ball size. For other species, they grow even faster. The one at the bottom of the panel took about 6 to 8 months just to cover the entire bare rock. So you can see that different species, they have different growth rates, and this is what we are all considering for our restoration projects. But when you look at it over time, the coral transplants eventually do contribute habitat and food for the marine organisms. So when you take all this into consideration, restoration is in fact a viable management option for Singapore's reefs. And what we are really learning here, we feel is applicable and relevant to reefs elsewhere. They are also experiencing high sedimentation and unstable substrates. So yeah, that's it for me. Thanks for listening.
Thank you, Lionel. So now let us move to Daisuke. So Daisuke started his marine research career in 2014 when he was hired for a coral restoration project with me in Tropical Marine Science Institute. Yeah. And then he developed his interest in fish ecology in Singapore's unique urban coastal environment and subsequently pursued his research in this topic. So anytime, Daisuke.
Sorry.
It's okay.
Okay. Good evening, everyone. Thanks, Sam, for the nice intro and edX for opportunity to share my research. So my name is Daisuke. I'm from a third-year PhD student from Experimental Marine Ecology Lab. So my PhD research is about this is what I call urban coastal fish ecology. And this is about studying the fish living in a human-dominated coastal environment. So in this case, I'm specifically looking at fish living in the seawall. So as Lionel talked a bit about coastal development, much of the Singapore coastline is made of seawall. So you can see these orange color coastline along the Singapore inside the Singapore map. This is where all the seawalls you can find. So seawalls actually very abundant features in our coastline, but we actually don't know very much about the marine life living in a seawall. And I was particularly interested in studying fish in this. So much of my research is based on scuba diving fieldwork. So I go diving and do the visual fish sensors in the southern island of Singapore. And first thing I started to look at is to see whether the seawalls can support the fish which are similar to those in a natural habitat. In this case, I'm looking at the seawall and the coral reef, which are located nearby. So there are a lot of fish which were found in these two habitats, but these two habitats actually, fundamentally, they were quite distinct. They had very distinct fish composition they supported. So this was largely because of the bottom substrate. The benthic composition of these two habitats are very different. So if you look at the seawall, this is what seawall look like. It's a bunch of rocks there, and the coral reef look like. So if you look at the seawall substrate, they have a lot of thin layer of these algae growing on the rock and not much else. Whereas coral reef has more diverse and high cover of corals and also other organisms that fish can hide and feed on. So that's why they're quite different. And I also observed a lot of fish behaviors and activities going on seawalls. For this, rather than doing a visual sense, I use a lot of GoPro cameras to observe these behavior. So I was particularly interested in the role of these algal turfs that as a food for the fish that is attracted to the seawall. So, and I took about 4 hours of video during the daytime and looked at fish feeding activity on the seawalls. And it's quite surprisingly, they have a lot more feeding activities on these algae going on seawall compared to the coral reefs. And this includes some of the important coral reef fish, things like parrotfish and rabbitfish here, and also the damselfish. So this kind of suggests that the food availability is quite one of the important factors that drive the fish community on the seawalls. Okay. Then I also look into explore some of the options to enhance fish diversity on the seawalls using the approach called ecological engineering. So ecological engineering is an intervention to enhance fish biodiversity on these urban structures without compromising their primary functions. So in this case, the seawall's primary function is to protect the shoreline against the erosion, wave action, and sea level rise. So this could be done by incorporating some habitat features that benefit the marine life into the construction process, or by adding some of the structures to the existing seawalls. So in my case, I use the second approach. So I have two experiments going on, and one on the top, I added some of the textured tiles on the seawalls to increase the surface area for the benthic organisms to settle and grow. So that provides more food for the fish and increase their biodiversity. And the other approach is in the other approach, I tested whether the addition of shelter space and shelter complexity can enhance the fish diversity. So with all these work, some of them are not finished yet because I'm still, doing PhD work. So with all these, I'm hoping that my research can improve our understanding of local fish diversity in Singapore, and also contribute to knowledge related to fish ecology in the urban setup. And I hope that helps to design more fish-friendly seawall ecological engineering interventions. And if I have time, I would like to share some interesting video of fish found in my fish surveys. So these are some sharks and rays that you probably don't encounter very often when you're diving in Singapore. So especially the one on the right bottom, I'm not sure if you can see, but it's a spotted eagle ray roaming around. So it's good to see these fish still, roaming around our local water rather than seeing them on the fish market or whatever. And I also have another fish which I want to introduce. So it's the honeyhead damsel. And this particular guy is quite good at tidying up their own territory, removing sediment and trash. So they do quite well, just like Maria Kondo's. I just call it Maricon fish as well. So, okay, this is all for me. Thank you.
Thank you, Daisuke. It's pretty cool, right? It's like building HDB for fish. Okay, so next up, we have Jenny. So Jenny is currently a final year PhD student. So for her thesis, she studied on the competitive interactions between corals and seaweed. So particularly on how seaweed may affect coral populations on Singapore reefs. Jenny, please.
Thanks, Sam, for the introduction, and thank you, edX, for giving us young marine researchers an opportunity to share about our work. So for my PhD, I focus on the competitive interactions between corals and macroalgae, or more commonly known as seaweed. For the past decades, competitive interactions between corals and macroalgae have become increasingly common occurrences on coral reefs. This is because coral cover worldwide is in decline because corals die due to global warming and ocean acidification. This opens up space for macroalgae to grow and become abundant. Increasing nutrient levels in the water due to pollution, as well as overfishing that reduce the number of fish that feed on the algae, also further promotes the proliferation of macroalgae on coral reefs. As a result, corals and macroalgae come into contact more frequently, and these interactions are likely to shape how benthic communities on coral reefs are structured. So for my research, I focus on how often do corals and macroalgae interact on reefs in Singapore, how these interactions vary temporally and spatially, and also look at mechanisms of how macroalgae may damage and harm corals, whether through physical means such as shading and abrasion, or through chemical means such as aminopathy, or whether the interactions are microbially mediated. I also look at what are the outcomes of these interactions and how these interactions may affect coral health, growth, or microbiome. So to answer some of my research questions, I do scuba diving to monitor and record the frequency of interactions between corals and macroalgae on Singapore's reef. I also conduct experiments underwater, such as the one here, to examine the effects of macroalgae chemicals on corals. I also did 10 experiments to mimic the interactions between corals and macroalgae to better understand how these interactions may affect coral health and microbiome. I also tested the effect of macroalgae chemicals on coral larvae to know whether or not macroalgae affect the early life history stages of corals. While the focus of my main research is mainly the competitive interactions between corals and macroalgae, I'm also interested in macroalgae taxonomy. Macroalgae is one of the more understudied groups in Singapore because it is hard to identify them to species level in the field. They come in various forms, colors, and texture. Most people will recognize macroalgae as either brown, so those like the first row are all the brown algae, or green algae, like the second row, or red algae. Although color may be misleading, such as the one like in the right-hand corner here, is actually a red algae, although it looks green. Macroalgae also have different textures, whether they are velvety, filamentous, spongy, or leathery. And while macroalgae may have bad reputation because they are known as the weeds in the ocean and they are harmful to corals, macroalgae actually have important roles in marine ecosystems. They are the primary producers, so they provide food for other fish and invertebrates. They also help in nutrient fixation and cycling and provide habitats for other small organisms. So while macroalgae may not be the most charismatic organisms on coral reef, it's important to study them given that they play important ecological roles in the marine ecosystems. That's all from me.
Thank you, Jenny, for your sharing. So next we have Samantha Lai. So Samantha is currently reading her PhD, and her thesis aims to investigate the potential of vegetative seagrass fragments as dispersal agent. And this will primarily be done by 3D hydrodynamic modeling. She also believes that scientists have a crucial role in ensuring environmental policies and laws are grounded in science. So as such, she is interested in applying her scientific training to help develop and apply effective environmental policies at both an international and national level. Samantha, please.
Thanks, Sam. Thanks for that introduction. And thank you, edX, for having me here tonight. Well, so my topic's about seagrass, and I guess I'm the first person who doesn't have to dive for her fieldwork. So before I even get stuck into my research, I want to talk about what seagrass is, because I realize a lot of people don't know what seagrass is. Number one, it is not seaweed. So if there's one thing you take away from my presentation today, is that seagrass is not seaweed. As seagrasses are actually marine plants, so they are real plants like the one that you find on land. It's just that they've evolved to live in the sea in subtidal and intertidal environments. So they like real plants, they have roots and rhizomes and xylem and phloem and all of that. But you don't get that in seaweeds, because seaweeds are undifferentiated mass of cells, essentially. So we find seagrasses in soft and sandy substrate. They can grow in both intertidal and subtidal environments. Singapore has 12 species, which is half the number found in the Indo-Pacific, and that's pretty impressive considering that we don't actually have a lot of seagrass meadows left. We have lost more than 45% of our seagrass, but we actually still have all the species that we've always had. So we haven't had any seagrass extinctions in Singapore yet. Seagrasses form large meadows, which are very important coastal habitats, but they are a bit less popular than coral reefs and mangroves, so people don't know about them. They have a bit of a charisma issue, but they're vitally important. They prevent coastal erosion because they do trap a lot of sediment, and they serve as a nursery habitat for a lot of marine species, especially those that live in coral reefs. They also sequester a lot of carbon in their sediment, and they actually do so at a rate that's higher than terrestrial rainforests. So they are really literally the rainforests of the sea, not coral reefs. But they are also very important for nutrient cycling, and they serve as food for important animals like dugongs and turtles. That's why dugongs are called sea cows, because they eat seagrass. And of course, they support other coastal ecosystems that are nearby, like mangroves and coral reefs. So my work focuses very much on the connectivity of seagrass meadows in Singapore. We have quite a few of them dotted all around offshore and on the mainland. And we want to know how well they're connected, whether they are seagrass fragments, which are bits of seagrass that break off and can actually re-establish elsewhere and grow elsewhere, or fruits, how far whether they can actually mix between meadows and transfer between meadows to help improve the genetic diversity. So to do that, I did, as Sam mentioned earlier, I did hydrodynamic modeling. So we use this thing called an agent-based model, in which we pretend we put into this program the behaviors of seagrass fragments and seagrass fruits, how they would behave in the under current conditions or wave conditions in different hydrodynamic conditions, and then we simulate how they would actually move in real life if you release them from different spots. So as you can see here in the screen, we can actually kind of predict where they'll move based on what we know about our currents and tides. So to do that, you obviously need to know a lot about how fragments and fruits behave in the water. And to do that, I actually had to do a whole bunch of experiments, which I did at the St. John's Island National Marine Lab, but also at the Royal Netherlands Institute for Sea Research in the Netherlands. And all these different experiments help inform the agent-based model to help me better predict where seagrass fragments and seagrass fruits would end up. So in the end, what we actually found was that seagrass connectivity in Singapore is very good. We found that in both the agent-based models and from our population genetic studies that our seagrasses actually do mix a lot between the different meadows. And it's not just within Singapore, they are connected to meadows in Malaysia and Indonesia as well. So if you can imagine fragments being released from this star, which is Labrador Park, they do end up as far down south as the Riau Islands. So there's a lot of regional connectivity because, well, obviously marine animals and marine plants don't recognize human man-made borders. So they do move all over the South China Sea. And obviously, I realized that we actually do have a lot of research gaps in Singapore about seagrasses, so we do need to focus future research on documenting the reproductive output of seagrasses so that we can better understand how they are moving around and how they are connecting between different meadows, not just in Singapore, but all over the region. So hopefully I've convinced you that seagrasses are important and they are interesting thing to study and not any less interesting than corals. Yes. So I'll hand it over back to you, Sam. Thank you.
Thank you, Samantha. It's funny how the coral people and the seagrass people are always competing.
Always fighting, right?
Okay, and always fighting. So
Yeah,
both are important and we need to say both, okay? Coral is a bit prettier, but okay, yay!
I can see where your allegiances lie.
Okay, so now we have the taxonomies. We have Nicholas. So Nicholas is trained as an invertebrate taxonomist while in the midst of writing his PhD. So very little is known about sea anemones in this region. So his research is centered on them. And I think I see a lot of questions on sea anemones already.
Oh dear.
And this has taken him across the globe in a goal to really identify and classify these animals accurately. Nicholas weaved both scientific data and historical records to construct identities of sea anemones, in many cases putting their face to floating names. Okay, so he also believed that without good species identification, there can be misinterpretation of biological data results that can hinder conservation efforts. So Nicholas, please.
Right. Yeah, thank you, ADEX, for having me here. And I'm going to talk to you about how I construct identities, putting names onto the animals that we see out there, and vice versa, putting faces onto the names that we know as scientists. But instead of going through all the science, today I just focus on one aspect, which is the historical data that I use to construct the identity. So you know what sea anemones are? They are related to jellyfish, they're cousins of the corals, and the one you see right in the middle that is staggering a little. Now that one, when we got it, it was this small, but it can grow up to the size of a typical coffee table. It is a host to clownfish. And they're really, really very pretty animals. Oh, sorry, it seems to be stuck. Yeah, they're really pretty. And many times, if you go out for your dives, if you go along the shore, along the rocky shore, you see this, hey, you'll be thinking, hey, what is this? Is there a name for this animal? And more often than not, I get questions like that asking me to identify the sea anemone that they see out in the field. But on the flip side, for us, we have a lot of scientific names that are floating. And when they're floating, meaning to say that we know their names, but we do not know how they look like. And they're all John Does and Jane Does, right? So that's my research, really, to link how the live animal looks like and what the name is. And we do that through collecting data in several ways. One of it is genetics, of course. And we synchronize that. It comes hand in hand to looking at the form of the animal and studying about its biology. Then we start to construct a bit of its identity. But intertwined tightly with that is how the species, how the animal was first defined, and that falls back onto history. So I have to look, and a lot of my colleagues have to look at historical artifacts, historical records, and historical accounts, or what we call descriptions of how the animal is first defined. And apart from going out to the field, apart from doing genetic lab work, a huge part of my PhD or my study, I traveled to different museums looking at specimens. And the specimens I look at, these are what we call, or what I call to you, the benchmark specimens, and what we scientists would call the type specimens. All these benchmark specimens have a special status because they hold that name alone. They have the special entitlement to hold that name alone. And going to look at the specimens gives us the starting point to help us to redefine what they are. And apart from looking at that, you know, in a perfect world, looking at that, it might help us to solve what they look like, you know, what names to put the pictures that you take of anemones to. But more or less, that's not often the case because some of the specimens get mixed up due to poor management or, you know, due to due to accidents. Some of them get lost, like some specimens got destroyed during the war. And some of them that remain may not be too helpful, like you see the microscope slides on the top right. Now, those slides alone cannot tell me how the entire anemone looks like. So we do our best. And we do our best, we have to go back to the original accounts, the descriptions, to try to step into the shoe of that scientist back then, to understand his psychology of how he defined that animal that we see today. But that's not an easy task either, because most of the time, they wrote really, really short paragraphs, and they wrote in a different language. I'm not fluent in Latin, I'm not fluent in German. So it takes a bit to translate and to understand the nuances that they are trying to convey. Some of them provide pictures, but more or less, most of the time, those pictures are not useful because they are over fanciful. Back then, they had to make a profit, right? So they make the pictures pretty so that people will buy the publications. And at the end of the day, like I said, you really have to do what you can. So we intertwine, we marry the two to synchronize them in such a way that at the end of the day, when we want to, and today in modern times, when we want to identify the anemone, there must be a standard, it must be standardized so that other scientists, other colleagues, other conservation people out there can find that identity objective and useful. And then we can start to plan, new conservation efforts, plan new studies to understand about our ecology, our biology out there. Case in point, the clownfish anemone. Before the 1980s, the taxonomy was a huge mess. The identities of all these anemones were not synchronized, not standardized. Then came in Daphne Fulton, back then Daphne Dunn. She wrote this one big study to synchronize their identities, and it's still being referenced today. It's still being used today because this, from her study, that is how people define what they are looking at, how they're protecting it, how they can study it further. There's over 200 citations of this paper alone that was published in the 80s, and it's still being used now. And on our side, on my project, one of the Jane does that we see very common along Singapore shores, it was first discovered in a museum as a forgotten specimen, and no one knew how it looked like in 1877. No one knew how it looked like alive. But after more than a century, we put a face to Jane Doe, and that's Phymanthus pinnulatus, a very pretty anemone that's symbiotic with anemone shrimps. And from there, we get to start, we take it a bit further, we study its reproductive cycle, and we kind of found in some ways that its reproduction coincides with the mass spawning periods around Singapore. So that's pretty cool. And with that, there's more to come. But I think the take-home message is, if you want to protect what you have, the first step to wisdom, well, supposedly Linnaeus said that, is to know things themselves. So yeah, thank you for listening.
Thank you, Nick.
Yeah.
And that is really a very, very nice quote. Okay, so next up, we're going to have Chi Kong. So Chi Kong is a part-time PhD student, and his day job is a research associate at the Tropical Marine Science Institute. And his current research interest is on the taxonomy of crustaceans. And he also taught me a little known creature, which is called 1098. Okay, he will tell you more about it. Go, Chi Kong.
Hey, hello everybody. I'm Chi Kong. So I'm a part-time student as well as the full-time staff at NUS. I'm based at the St. John's Island National Marine Lab, which is located on the beautiful island at the south of Singapore. You can see behind me, it's sunny. And despite Singapore being greatly urbanized, it still offers a huge diversity of marine life. So for example, there's still snakes in the mangroves of Sungai Buloh. There are still limpets on the rocky shores of St. John's Island. There are snobby sea stars at the north of Protocong on the reef flags, and there's lots of sea urchins and symbiotic worms for you to study on their symbiosis relationship. And there's some interesting flatworms. In fact, there are, I think, many different kinds of species of beautiful flatworms that we still don't know their names, and there's still so much diversity out there in Singapore waters that we can study on. And if you're feeling gloomy about our current situation of our environment, I think rest assured that there's still a lot to offer. So if you are a student looking for study subjects, I think there's no limits to it. So so my current research interest is termites. I learned something from Sam this morning. She wrote down the notes of how I pronounce termites as 1098. I think it's pretty cool. And termites are very similar to isopods and amphipods, which you might be more familiar with. And the difference between these termites and other small crustaceans that you might encounter in our local waters is the chela. Okay, so one tip is to remember the chela. So these small crustaceans are usually about 2 mm in length and can be up to maybe 5 cm the most. And sometimes they are really small, like 0.5 mm. And when you talk about crustaceans, people tend to ask, can eat or not, or whether this animal can harm me or not. But we still don't know much about the biology. And as far as I know, we cannot eat them. Nobody eats them. And they don't cause any harm to people. They are not parasites like some isopods or other crustaceans. So there's still a huge diversity of termites waiting to be discovered. Experts have estimated that there might be up to 50,000 species worldwide, and there's only 1,500 species being described. So there's like maybe 2-3% out of what we think there is. So there's still a lot to be discovered. And recently, I and my colleagues we described 3 new species of termites. And this is just a tip of the iceberg. We still have lots of material to process. And in Singapore alone, we found at least 25 species. And there could be more because we haven't even actively go and search for them. And these are just based on what we have found deposited in the museums and our colleagues who hand over some of their material. Then we look through them based on the morphology and maybe based on molecular data in future. We already identified at least 25 species of termites in Singapore waters. So how do we distinguish the different species? So termites, like other arthropods, they have jointed limbs. So they have lots of antenna and what the walking legs we call pteropods and swimming legs and of course the chela that I mentioned just now. So and the mouth parts. So we have to dissect the animal into at least 18 parts, and every single part of the animal we have to look carefully under powerful microscopes because they are so small, and every part could be less than a mm. And for each part, we have to look at the small hair and even the morphometrics of the parts. And even if you look at the picture here, each leg is divided into even smaller joints. So as a taxonomist, I have to examine each part very carefully to compare with similar looking animals, so that to be really sure that they are of different species. Yeah. So that's all I have. Thank you.
Thank you, Chikong. That's super interesting. So now everybody will remember 1098. Okay, to round out the presentations, we have Aidan. So Aidan is our last panelist, and his research interest lies in molecular ecology, with focus on the applications of animal environmental DNA, or short form is eDNA, and DNA sequencing. So much of his work involved the detection and biomonitoring of species to better understand marine biodiversity in Singapore. So,
Chikong, you need to turn off your screen. Yeah. Thank you.
Okay, so yeah, let's go.
All right. Okay, good evening, and thank you ADEX for having me here today. I'll be sharing on how we use DNA methods, particularly environmental DNA, to better understand marine biodiversity in Singapore. So to first answer the basic question, what is eDNA? So essentially, is DNA released by animals into the environment, such as through the shedding of skin cells, mucus, feces. These methods involve the recovery and detection of shedded genetic material from environmental samples, for example, from collected waters in this case. So why the use of eDNA has grown popular recently is that it is a non-invasive sampling alternative that allows us to know if the animal was present without the need for direct capture. So it also offers many other advantages to complement conventional surveys, such as the reduction of manpower, cost, and time needed for traditional sampling. And most importantly, it gives us the ability to bypass typical field challenges, where we can now obtain information from sites that are hard to monitor, such as those with strong currents or poor visibility, like in Singapore. You can see here in the photo on the bottom right, we get an average of 1 to 2 meter water visibility, making it tough to monitor animals or even to spot pelagic species. So my work as a PhD student generally involves molecular ecology in the marine environment, with special focus on devising eDNA methods for species detection, looking for invasive and rare species. Also, to be able to use eDNA signals for differentiating between habitats, and last of all, for biomonitoring key reef species, which will ultimately have greater conservation implications. What my team and I have done in Singapore so far is the successful application of eDNA methods to tell habitats apart. We have sampled water and recovered eDNA from 8 sites in the southern islands of Singapore, and we managed to find over 500 animal species. We also found that each habitat type is made up of a unique suite of animal signals, which goes to show that if we were given a sample of water without being informed on the collection source, but with the power of eDNA, we will be able to work backwards and determine where the water was taken from, be it from a seagrass environment, mangrove, an intertidal or subtidal reef. Other interesting results of this study includes the detection of invasive species, which has further implications for our management strategies, as well as rarely seen animals like sharks, rays, dolphins, and turtles in Singapore. We further explored other applications using eDNA to biomonitor diversity here, such as to use it as a less invasive monitoring alternative to monitor annual coral spawning events. This ongoing work with eDNA can provide us with more information to study this rare phenomena that only offers us a very narrow window of opportunity to dive and observe. What we have so far is the ability to detect some coral species that have spawned during the expected window without having to dive and physically observe the actual spawning. The results are very promising. We do find that coral eDNA signals increase extremely sharply during the predicted coral spawning window, which suggests its potential in complementing traditional surveys. Additionally, we have used eDNA to detect animals that are rarely seen in Singapore, particularly sharks and rays. We occasionally see about 7 species of sharks and rays compiled from online photos over the last 20 years. But with eDNA, we found 16 species in Singapore. And based on the results patterns, they seem to be more common than typically observed. So some reasons we can suggest as to why we don't see them often could be due to the poor water visibility. And also, elasmobranchs, they generally tend to avoid human contact. Regardless, we found 2 times more species than usually seen. And these are either new records for Singapore or a species of conservation concern. This is still ongoing work. We are still working to uncover more unseen species, and also to determine if any of those that were recorded in the past have actually become absent today due to environmental changes. So to end, I would like to emphasize that eDNA is a very powerful molecular tool, and its application here complements conventional methods to help us better understand Singapore's marine biodiversity. Ultimately, our combined effort between work done with eDNA and traditional surveys aim to contribute to more meaningful implications for biomonitoring and conservation of animals found in Singapore's waters. And with that, I will end my presentation. Thank you for your attention.
Thank you, Aidan, and thank you all panelists for your presentation. So now you can take a little break while we find some questions from the audience for you all. Okay, so I think the first question goes to Daisuke. Okay, so what is the percentage of algae turf coverage on your coral reefs versus the coverage on seawalls? And did this influence your fish count and data?
Yeah, so from my recent surveys I did last year, so I compare the seawall sites and coral reef site, and we did the fish survey and also benthic survey. So from there, about algae cover and seawall is about 80 to 100%, and the coral reefs, the algae cover is about 40 to 50%. So that would definitely affect what kind of fish are associated with these habitats, and probably the number of fish which are associated with algae is a lot more in seawall that way. But these surveys are done in a standardized area, so there wouldn't be any bias towards from one to another in terms of number, in terms of the count of the fish. Yeah.
Okay, thank you. So another question for you. Super interesting stuff. Okay, so Lala asked this question and she's not sure if you mentioned this, but she's curious if there's any reef near the seawall that you have observed. Could a near reef affect the abundance of reef fish at the seawall?
Yes, I think so. So I, again, like I said earlier in the survey, so the seawalls, on the seawall that is near the coral reef might have some fish that are associated with those coral reefs. So the parrotfish is typically found on coral reefs, and they might come and swim by the seawalls, which are located nearby. So, and that's why we found a lot of parrotfish actually probably feeding on the algae in the seawalls. But if you look at some coral-dependent fish, something like butterfly fish, which is found on a coral-rich environment, also feed on the corals, you don't see these fish on the seawalls very often. So yeah, okay.
Okay, thank you. So next one, we're gonna ask Samantha. So this is by Pei Yan. So is there a possibility of new seagrass meadows forming in locations without seagrass?
Yes, of course. So, I mean, well, Pei Yan should be familiar, but we do have a lot of seagrass meadows on reclaimed shores. So, East Coast and Tanah Merah and Changi, they're all areas that have been reclaimed in the 60s or the 70s. And as long as there's the conditions are right, you have the right substrate, you have the right hydrodynamics, and you have, more importantly, a source of proper use. So, the fragments of fruits that can land there, then there's no reason why a new meadow can't form. So, I mean, our reclaimed land meadows are clearly an example of how this can happen.
Samantha, in your presentation, you mentioned something about rhizome and you showed video about how this seagrass dislodge under strong tidal wave or waves.
Yeah.
So I believe there is one related question is from Pei Yan as well. Is there a possibility of new seagrass meadow forming in location without seagrass? So I guess that video actually could you share more insights? So basically what happened is I believe rhizomes need to be attached somehow or rather to the substrates. So how far can they travel? And if they, after traveling, if they don't settle down, can the rhizome be grown suspended in waters?
So, okay, several parts to break down. Seagrasses can reproduce by different ways. So what we call asexually, which is by fragments breaking off and then being transported elsewhere. So they are like land plants which can, because they are clonal, they can be, you know, parts of them can break off, and you just need preferably some meristematic tissue. But okay, if you have rhizomes with healthy roots and shoots that with leaves, usually they can, in the right conditions, settle and establish elsewhere. The chances of that happening in the wild, in nature, is actually quite low because it's very unlikely to actually land in the area with good substrate and with the right hydrodynamic conditions, which are slow enough for the plant to settle and establish. So there are very low chances of that happening, but you do have a lot of fragments. So that's that increases the possibility of that happening. They can also reproduce and disperse by fruits because a lot of seagrasses produce fruits that can float. So these can actually travel very far. So for my models, you can find that the fragments and the fruits can travel up to 30, 40 kilometers, no problem. So which is why they are entering Malaysian and Indonesian waters, like far outside of Singapore waters. Yeah. So I mean, that's one of the ways that they disperse, basically, for seagrasses. Yeah, and they can definitely establish in areas where there's no seagrass if the conditions are right.
So that leads to my next question. So am I right to say that seagrass can grow on fresh water or even near mangroves? I know seagrass grow near mangroves, but how about freshwater, like lakes and stuff?
No. So, okay, strictly no. So if it's a seagrass, it grows in saline water. Brackish water, sometimes there are some species that can grow there, but mostly in saline water. So you have them in marine environments. They're the only true marine plants, so they are completely saline. Yeah. There are some people, okay, let's not get into that competition because it's a discussion for taxonomists whether they're actually seagrasses or not. But the strict definition is that no, it has to grow in marine, in salt water. Yeah.
Okay, thanks Samantha for the answers.
Okay, so next questions for Nick. So this is from Pei Yan. So how do you make use of specimens in a museum? So to know more about the specimen and what do you do with the specimen, especially when you don't want to damage such precious specimen?
Unmute.
Nick, I think you're on mute.
Oh, yeah, there you go. Unmute myself. So, unfortunately, for sea anemone taxonomy, as Pei Yan well knows, you do need to take a bit of a tissue sample to sample for its stingers from its cells. And you do need to slice it up to look at the internal morphology of it. Actually, to better express this, can I share a screen? Yeah, am I allowed to share a screen?
Yeah, sure.
Yeah.
Go ahead.
And there you go. So in pictures C and D, D is a live specimen that we got, and C is the museum specimen. So on the first step that we take is to try to see similarities to what we have, what we see out there to the original museum specimens. And usually if you want to do sampling on the specimen, we have to ask the museum curators for permission or look for slides that have already been prepared. And then if they say yes, we can go ahead with the sampling to look at the stingers. The stingers you see there, those are things when you touch the anemone, it stings you from letters A to N. And then we look at the muscles of the sea anemone. And these all comes from the original benchmark specimens that help us define what they are. Are they the same? Are these reported in the original accounts of the species? Do they differ? If they differ, that means that the so-called benchmark species is not really the benchmark species at all. Someone might have messed up somewhere, or maybe they might have overlooked these features. So in short, Pei Yan's question, that is how we use the benchmark or type specimens, the museum specimens as we call it. We try our best not to damage it over time and or take too much of it over time. And that's about it. And if we do take pieces from it over time, we actually have to put a label down into the jar to say that Nicholas has cut a piece out of this, and the label stays in the jar like forever. And that gives people a perception, at least they know who worked on it, and so on and so forth. Yeah.
Right, Nicholas, talking about sea anemone and our topic today in regards to marine biodiversity, can I safely say that most or all sea anemone contribute towards the beneficial ecosystem for marine biodiversity?
On what basis do you say that?
So, like, for example, they filter out toxin from seawater, and then, I mean, I'm not a subject expert, man.
Well,
I'm not subject expert. So, this question, curiosity.
Well, no, I think every single organism plays an important role in the ecosystem. Every one of them do. But it really boils down to perspective of what one deems as important. I think case in point, maybe the magnificent anemone, the big purple one you see with the clownfish. And for the longest time, I think it's seen as something that is really, really important to the environment because it's symbiotic to the clownfish. But I mean, from my own fieldwork perspective, I see them as kind of like pests because they're really opportunistic, and they really quickly colonize an area where other say more supposedly more important species can colonize, like corals or seagrass. And there's, unfortunately for sea anemones, there's still not much research focused on them to really understand about the ecology, save for the few clownfish anemones out there. But this is my own perception. Yes, they're important. Everything is important. At juvenile stages, there can be plankton for food, as with many others like fishes and all that. But to what extent, it all boils down to perspective, really, and how you market it to gain the funding to do research on them. Yeah.
Nice. Thanks, Nicholas, for enlightening me.
Jaded, not quite.
Okay, so the next one, we have questions on 1098. So we're going to ask Chico, what is the role of 1098 in an ecosystem?
Based on the limited literature that we have, we know that they are prey to a lot of fishes. For example, those benthic feeding fishes like muscapers. In fact, I have one colleague who found once a night in the stomach of a muscaper. So then, and also in other parts of the world, you find a lot of reports of fish having termites in their gut contents. And as a consumer or predator, they are found to feed on algae and even the seeds of seagrass. Sorry, Sam. Yeah. So then, and also in deep sea, I think they believe to feed on foramiferans. I mean, forams, those small hard shell or soft-bodied protists. Then, because they are fully benthic animals, meaning they reside on the seafloor and they build so-called tunnels, and then they surround themselves with a tube using the sediment from the surrounding. So in that sense, maybe they add like earthworms, like do some like bring up organic material that would otherwise be trapped inside to the surface. So these are just some of the roles that I can think of right now.
Right. So, Chikong, the Priscilla asked whether we can find 1098 in Singapore waters.
Yeah.
And whilst answering, before you attend to that question, I have hold on. I have Chee Hoon from Friends of Marine Park Dive Team sharing a picture. I'll just share screen quickly of 1098. So in Hantu waters, to answer you, Priscilla. So Chico, have you seen something like that in Singapore waters?
This is Chee Hoon, right?
Yeah,
you see Chee Hoon?
Yeah.
I saw this picture by her. It's a very rare photograph because for some reason, okay, not for some reason, because they are small. So divers tend not to take photographs of Tanaics, and they are quite colorless and nondescript and not very pretty in the sense. So I'm very happy and surprised at the same time that when somebody took the photograph of a Tanai. So just these are Tanai. Yeah, so of those species that they have modified the first walking legs as a brush. So this is highly modified. We still don't know yet what's the specific function of it, but because it has many sensory hair at the tip of the first pair of walking legs, so maybe it's aid in some kind of feeding purposes. So to really answer that question, we need to do some kind of lab work, and then observe them under controlled conditions.
All right. So I hope that answer your question, Pris. So Sam, shall we go on to the next question?
Yes, and we also have a lot of questions on eDNA. So this is for Aidan. So this question is from Christina, and she asked if sampling is conducted at different water depths, and are benthic species can also be detected through eDNA?
So yes, the way we do eDNA sampling in Singapore is we sample from both the shallow and the deep water. We consider 10 meters as deep water because our reefs reach up to maybe 6 to 8 meters in depth. So we do sample at both depths because we, in the literature as well, there's differences in community assemblages from different water samples collected at different depths. And regarding benthic species, yes, we do detect benthic species, and usually intuitively you find them in the deep water samples. However, I just want to caution that eDNA sampling is not that straightforward. So the detection is actually a very complex process. Does not mean that if you sample somewhere near the animal, you will always get a positive detection, because there are many other factors that you need to consider, such as life history, traits, the rates of shedding. Like if the animal is like a crab, it has exoskeleton, it doesn't shed that much DNA into the water compared to a fish that excretes along the way or has a lot of mucus being shed off. So that's about it.
Okay, and thank you. And also to add on this question from Cui Qing. So they asked whether water flow will it affect the reliability of eDNA collected, and how long can eDNA survive in the water?
So water flow, we can't completely exclude. We can't completely say that signals from one site, let's say Kusu and Lazarus, they're very close. We can't say that, this signal is exclusively from Kusu, because marine habitats in Singapore are all connected, you see. But in that regard, if you're just looking at specific habitats, we can look for animal species that are specifically found in certain habitats, and then we can use them as a marker to distinguish habitats, the habitat signals. And so water flow to a certain extent can sort of confirm your results, but we have to treat that with caution. That's why we have to look into every single animal species that we detect and try to make sense of it if we want to look at habitat discrimination. So how long can eDNA survive? So basically in Singapore, it has not yet been tested, but there are other studies that showed that in the tropics, eDNA does not have such a long half-life. It doesn't survive that long because we are in the tropics, we are near the equator, there's lots of ultraviolet rays from the sun, and that is very bad for DNA fragments. It breaks down DNA fragments, and it will cause you to not be able to detect species that after a long time. So in that regard, it's very real time, you see. And also there's also the factor of heat. We are generally hotter, our climate, as well as because we are having high rates of sedimentation and all that, we have lots of humic acids, and these are numerous factors that will break down DNA fragments. So I would actually trust that the DNA signals that we detect from the samples are actually very quite substantially real time. Yeah.
Thank you. I think Carter has a question.
I have a question for Jenny though. Jenny, as you are doing your thesis, you mentioned about competitive interaction. Can they coexist at all without fighting? I'm talking about corals and seaweeds.
So if not fight, if you're saying that they are not fighting, that's known as stand-off, but that's also it's not good, because if not grow, that means that both corals and macroalgae are not growing, which is not their optimum health. But it's also
sorry, continue, Jenny.
So, but not all corals are susceptible to macroalgae, and not all macroalgae are harmful to corals. So some macroalgae are more damaging, like some chemically rich macroalgae are more potent to corals, and some corals are more susceptible to macroalgae damage. So depending on the species. So
for the healthy growth of both to coexist near each other near enough, then is it right to say that we need marine species to eat them up? I mean, turtles eat seaweed, right? And then some fish species eats some of the hard corals that they ingest. So, I mean,
because for the competitive interactions between corals and seaweed, space is the limiting factor. So both corals and seaweed, they need space to grow. So that when they come into contact, when they are just neighbors to each other, if one needs to grow, then one needs to be the winner of this interaction.
I hear you. Thank you, Jenny.
Okay.
Thank you, Jenny. So we have a question from Lionel. So looking at Singapore's corals, right? How will you compare that to the region? Are they doing okay? Or is there any special corals that we find in Singapore?
If you're talking about species richness wise, Singapore has historically recorded more than 250 species. Everyone knows that number. And I think for a small country like ours, it's quite good. If you look at the South China Sea region, they have recorded about 570 plus species. So it's quite a good number for Singapore. In terms of reef health, of course, we are degraded. But that doesn't mean that we cannot find new things too. As late as last year, we have found a new record of hard coral. It has always been there. It just went unnoticed by a lot of people, and we were lucky to find it. So every now and then, new records show up, and that's why Singapore's waters surprises us and keeps us interested in working in this field.
Thank you. Okay, so we have a question to all panelists, so you can take turn to answer. So this question is from Desmond. Okay, so his question is, although the marine environment provides us with a plethora of goods and ecosystem services, but in a Singapore context, we don't necessarily receive all this in return. So why should we protect marine biodiversity in Singapore? And how do you convince the public that this is necessary? Anyone can start first.
Okay, maybe I'll go first. If I take the easy answer, then the rest of them have to do the hard ones, right?
Yeah, that's a good one.
Good strategy. Well, okay, I don't quite agree that the public doesn't get any of the goods and services. Maybe they're not aware of the goods and services that they receive. So I definitely do think that a lot of, you have things like carbon sequestration, which is not something that you can see or take or eat or feel, but it does do something good for your environment. So there's that as well. Water quality and things like that, you don't really appreciate it until it's gone. So, but I think one important thing is also that these ecosystems and these habitats and these biodiversity, they are all part of our natural heritage. So they have been around even before any of us, before any humans have been around. So if we won't think of, demolishing our old buildings, our Victoria Concert Hall, our Parliament building, why would we think of destroying habitats that have been around much longer than that? So that just doesn't make sense to me. So I think it's a matter of awareness, not being aware of that being part of your heritage, that being part of your history and your culture, and that we need to convince people about. So I think it's a matter of education, it's a matter of awareness.
Yeah, I think it's a good answer. Anyone else would like to add on? Daisuke?
Yeah, I'll add on.
Talk a bit as well then. I think I agree with Sam on like the parts she talked about, and especially the natural heritage. Singapore, well, before the settlement and Singapore settlement with Raffle Cames, and it used to be a fishing village, and the 20th century, early 20th century, people were still living in some of the offshore islands, and they were making living by fishing, and and also ornamental fishing also, and they're appreciating all these natural heritage. And I think our future generation, from Japan, but our future generation here would deserve to see all these to natural heritage, and also understand the history of these things. And I also think Singapore is one of the leading examples of these urbanization, and which coexist with the natural diverse natural habitats and ecosystems. So, not only Singapore, but at the regional significance, if we can protect these natural habitats and the animal living there, and as well we do the sustainable development. And other Southeast Asian countries like Thailand, Indonesia, Malaysia, other countries are also, some cities are quite highly developing, and they also want to seek out for a solution to sustainably develop while protecting the biodiversity there. So I think there is some significant in the regional wide as well. So that's my point.
Wait, Chico, you want to add something?
Yeah. I think even though the Singapore Biodiversity is still very limited, like a lot of places have been like with so little mangroves, coral reefs, and other natural places gone. But I think there's still a lot to offer to the public. Like even if you go to those small walks in Chijawa, or in the St. John's Island or Sisters Island, there's still so much for people to keep interested in nature. Like recently, right, there's still like a lot of turtle eggs being found in Spruce Park, or hornbills coming back. Not only marine biodiversity, but biodiversity in Singapore in general. I've seen a lot of people get excited when they see that all these animals are coming back and in their own backyard. I think it's something that we can call our own. It's maybe we not have the Serengetis or the Great Barrier Reefs, but I think we need something that we can be proud of, and it's also contribute to the Singapore story, right? Singapore, we talk about surviving against all odds. So in Singapore context, even though we are a highly urbanized, highly industrialized country, we have almost surrounded by artificial seawalls and whatnot. We still maintain a high level of biodiversity that we can be proud of. So I think in that sense, in terms of natural heritage, I think people can be convinced to conserve what little left we have.
Okay, thank you. So that was interesting. And then, okay, so I'm going to have a last question. So this is to all panelists also. So for your research, what are some of the barriers between implementing environmental research into policies and public education, and how do you overcome them? Maybe Lionel?
So for mine, I'll answer in context of the reef restoration work that we do, and Sam is a part of our small team. So we understand that when we do reef restoration, the corals don't grow that fast, and in 1 month you see the coral transplants doing very well, and next month you go back, you see them being knocked over or being strangled by, say, fishing lines, and that gets us a bit depressed. So these are some of the challenges, and we are hoping to convey these challenges to the general public. We understand that the sea is a shared space, but we also want to know how the public thinks regarding these issues, so we can all work together to find a best solution forward.
Thank you. Anyone else? Nicholas, for education side, maybe?
Well, right now I'm in this headspace of trying to write out my thesis, and so everything's focused on taxonomy. I'm just going to go through that. I guess one of the barriers of bringing across taxonomy as an important science, among many of my peers, not you guys that are on the panel right now, but many of my peers and ex-colleagues, when you talk to them about taxonomy, there's always this perception that it is not a very applicable science. It's frivolous because they're just classifying things. There's no real application to it. And that translates into many different things. Like, it's really, I might be wrong, but it's sometimes very difficult to try to secure a grant, and or try to write a grant to state that taxonomy is important, unless you market it in such a way that it saves the world, so to speak. But that is, in my opinion, that is not really the point, because at the end of the day, you do need a good foundation for new science to build up, for new constructive ideas to come about. And sometimes the results with taxonomy itself is not instant. It feels like everyone wants an instant solution to solving problems, but sometimes it's not. So that's one of the barriers. I'm still trying to figure it out, and it's a bit frustrating. And yeah. And when it comes to education per se, working with the young ones, speaking to young ones, everyone, they know the importance of ecology, of conservation work. But when it comes to identifying creatures, it's something that's not quite there yet. They're more interested in foreign things than those on their local shores, really. So it boils down to really pushing out public education efforts. Yeah.
Thank you.
Yeah.
Maybe one last panelist can share his or her thoughts on this.
Maybe I could add in. So I definitely agree with Nick that there's a huge barrier to education, in that it's not the large part of like ecology, biodiversity, it's not informal education. So I think that's what needs to be done. Like you need to inculcate that at a young age and make our youngsters aware of the biodiversity that we have and to appreciate the natural heritage, because only then can you really, that goes back to answering Desmond's first question, which is, how do people, what value is it to them? Because if they're not educated about it, then they don't experience it in their lives, and then there's no value to them. And then that's how these habitats get lost, right? So I think we need to definitely push for it to go into our curriculum in some way, if not in the formal curriculum, then at least in terms of experiential learning, as part of your outdoor education. Instead of going to do team building and like climbing log poles or whatever, you bring them out to a mangrove and teach them survival skills or teach them about the environment. So I think there definitely needs to be a shift in focus in terms of our education. And I think with climate change being such a big issue, more and more environmental education is becoming important and being seen as important. So I think we do see that change. We're starting to see that shift slowly happening and that barrier is slowly going down, but I think there's still a long way to go. Yeah, thank you.
Thank you, Samantha. So to round up, I think it's really important, like what Penny has said, education in terms of whether it's formal or, even simply you start from home. So you can just think of, maybe you can bring your families after the COVID situation when you can all go out. Just take them to the nature, close to the sea, and really try to explore or rediscover what we have in Singapore. Okay, so thank you, panelists, for your insights and your time. So here's something to sum up today's panel. So I adapted from Nick's final slide. So if you can't love it, you can't love it until you know it. You can't protect it until you understand it, right? So these works of our local scientists, they don't not only contribute to advanced marine science, but some of their works are also used to formulate different strategies for species conservation and coastal management. So of course, scientists, we cannot do this alone, and we need all the help from you guys to help safeguard our marine biodiversity. So once again, thank you for all your sharing and joining me on this panel. So we have Lionel, Nicholas, Aiden, Jenny, Daisuke, Samantha, and Chikong from NUS, Tropical Marine Science Institute. And we wish all of you the best for your PhD journey, and we hope to see more of you guys in the near future. Okay, and thank you to all our friends who have stayed with us throughout the session. So thank you, and we will see you soon.
Thank you, panelists. Thanks for all your time, and hope to